And I'm excited to present our next speaker, Dr. Philip Horner. Dr. Horner is the scientific director of the Center for Neuro-restoration at Houston Methodist. He received his PhD at the Ohio State University and served on the faculty of the Department of Neurosciences at the University of Washington before joining Houston Methodist. Dr. Horner is going to be speaking this morning on the science underlying the intervention. Phil.
Thank you. It's a pleasure to be here. I'm going to give you the bench perspective on what I think are some exciting technologies that will be applied to stroke, in the sphere of regenerative medicine. So the bulk of my talk is really going to focus on neuromodulation or electrical stimulation, because I think that in addition to rehabilitation for the stroke patient, electrical stimulation is really the next frontier, but there's a lot for us to do and learn. So I'm going to give you more of the bench perspective, though, of where that technology can take us, and then I'll end with a little bit of a futuristic, not that futuristic, I think near future technology that I think will be used by the surgeon to replace circuitry in the stroke patient. So just a real quick acknowledgement, a lot of the data I'll show you is from the Horner Lab, the group that you see here. So my specialty is really spinal cord injury, but one can think about spinal cord injury as a stroke of the spinal cord in many regards. It's a functional injury to the vasculature of the spinal cord, and the spinal cord vasculature is really a watershed. So when you have an injury, you end up with a large spreading lesion, not unlike that which you see in stroke. Most of what I'm going to talk to you about is, actually, I don't know. Okay. So what I'm really going to talk to you about is this pathway, which is the cortical spinal pathway. So I don't know what I just did there. I apologize. I thought that was the pointer, and I'm not pointing. So let's, oh golly. Okay. So if you will, I'm going to talk about the cortical spinal pathway, which is depicted on your right there. That's the pink traces you see. So the cortical spinal tract is the primary motor system for motor behavior in humans, particularly volitional motor control of the hands and the upper limb. Okay. Should work now. Okay. Yes, very good. Okay, thank you very much. So, a lot of what I'm going to describe to you, it really doesn't matter if the injury is here to blood extravasation or if it's down here. What we're trying to do is reconnect the brain's volitional control for motor systems. And so this is a common problem in stroke, this injury to the cortical spinal tract. And one thing we know, just like spinal cord injury, we know that the large majority of patients have some spared fibers. So we rarely see a complete lesion of the cortical spinal tract. We see some sparing. And the question is, how do you bring that system back online? One of the challenges we have is obviously a very complex system. There are billions of neurons in the brain, and there are trillions of connections, and the cortical spinal tract is a very complex system. So how do we bring it online? The spared pathways, and how do we give those spared pathways an increased ability to affect the patient's volitional control? Well, one of the primary things that I think we're already starting to see in rehabilitative care is the addition of electrical stimulation, and that can be functional electrical stimulation of the muscle, that can be transcranial stimulation using magnetic systems, it can be wires in the cortex, or as Dr. Britz alluded to, multi-electrode array systems, implants, hardware implanted into the cortex. I'm going to touch on all those a little bit. But the main theory, I'm going to get into several theories, because I think if we need, one of the big challenges we have is we don't really know how neurostimulation works. It's kind of surprising to see a technology. I think it's one of the few technologies that's really starting to advance heavy into the clinic and into rehabilitative care networks, but we don't have really even a novel or a sort of a nascent understanding of how it works. So one of the reasons electrical stimulation was first applied in animals was based on the theory you see here. It's called Hebbian theory, and that's the predominant theory, although we don't know, there's not a lot of proof that this theory is borne out for electrical stimulation, either in animal models or in humans. But the theory is simply neurons that fire together wire together. And you see a network of neurons here. There's a big neuron A, and neuron A, you can see the little firing pattern. So you see the little ticks just below neuron A. That's the way that neuron likes to communicate. And you see two neurons B and C. Neuron B has the same communication pattern. So according to Hebbian theory, proposed by Donald Hebb, B and A want to wire together because they're firing in a similar frequency and pattern. And then neuron C fires at a different tune, and it doesn't hook up. So that's what we're trying to do in patients. We're stimulating regions of the cortex or stimulating regions of the midbrain or spinal cord during active behaviors, and the idea is to try to create Hebbian plasticity, so to create new connections between those connections that were lost. Now, just to give you a flavor that of how early our understanding of how this works, we know that there's a neural code, meaning that we know that neurons communicate. There's a variety of different types of neurons. There are obviously motor centric neurons or motor neurons. There are sensory neurons. There are neurons that are specialized for the sensation of pain. There are so-called interneurons, which are sort of integrating information from the sensory system and coordinating the output to the motor systems. And all of these systems really communicate in different ways. One of the simplest ways to think about it is our sensory systems usually fire at very high frequencies, 100 hertz, for example. Our motor systems usually are active at a much lower frequency, 2 to 10 hertz. So we don't yet know whether we can take advantage of those the language of these different systems really to increase the potentiality of electrical stimulation. If we look down below, therapeutic approaches, we're essentially limited to two approaches you see clinically. One is very high frequency stimulation, which is used for the amelioration of pain after neural injury, or a sort of intermediate range, 20 to 40 hertz is what is commonly thought. That's the frequency with which we get neuroplasticity to start to be awakened in neural systems. But just it's sort of a primer that we haven't really explored. Fundamentally, we haven't explored, even in animals yet, all of the different potential frequencies that we could use to promote plasticity. But at its very basis, in animals and clinically, 20 to 40 hertz seems to be this magic range in which we start to see plasticity. So I'm going to show you an example of how this can work in the concept of electrical stimulation to promote Hebbian plasticity and therefore recovery. So I'm going to show you a rodent model of spinal cord injury. This is a cervical model of spinal cord injury. So we've disrupted the descending input from the cortex to the spinal cord. And I'm going to show you one example, but we can get recovery of function in this animal by three ways. You can stimulate in the cortex, which is depicted here, during an active behavior, or you can stimulate just below the injury in the spinal cord, which is off to the right there, where you see wires have been implanted into the spinal cord. Okay, I've turned on a light show. I'm gonna not use the pointer again, I promise. So it doesn't seem to work for me. But on the right, you can see there are penetrating wires in the gray matter, so in the motor pools. The other way you can do this, which other labs have shown, is you can stimulate on the muscle. So you can do muscle-activated stimulation. So you measure the intent of the animal and then stimulate the forelimb musculature, and that can improve the behavior. So I'm going to show you this data. So this is data using a spinal stimulator. So this is a rat that had been injured in the cervical spinal cord. This is what we call a skilled motor test for a rat. Has to reach through a gap. Before the injury, they would have lost the predominant function of their hand, the ability to grasp, pronate and supinate. And graphically, it's represented on the left, the improvement this animal and other animals achieve just by stimulation. So the functional recovery occurs over about 6 weeks in a rat, and after that point you can remove the wires. This is permanent plasticity. So this is a really exciting result, but of course we can control a lot of the model system in rodents that we can in humans, and it's much more complex to translate this. But we are starting to translate this. So this is I got to go back. Should be a movie here. There it goes. So this is work by our very own Dmitry Sayenko. So this is using a variety of techniques, either a multi-electrode array that's placed on the epidural surface in the lumbar spinal cord, or transcutaneous, so stimulators placed on the skin. And what he's been able to show is you can take a similar technology that we've shown in rodents and other labs have shown in rodents. If you stimulate the lumbar spinal cord and combine that with, this is in a paraplegic patient, if you combine that with standing training, so putting the patient in a standing frame and then stimulating just enough, so basically you're just bringing central pattern generators online, you can get the patients to regain the ability to stand independently. And on the right, you see patient is also learning to locomote. So these are significant improvements in function. They're not fulminant regeneration or fulminant recovery, but they're significant. So can we apply this to the stroke patient? We're starting to see this technology applied to the stroke patient. And I think we're going to see this broadening. There's a little more complexity to the stroke patient because we have multiple pathways that we know are disrupted. So it's harder to isolate exactly where we need to stimulate. But we're starting to see this penetrate into the clinic, and I think we're going to see an increase in its application. So some of the questions, of course, for the stroke patient are, do we do this invasively or non-invasively? Meaning, it's probably going to be tailored to each individual patient. So do we want to use transcranial stimulation? Is the lesion more complex? Do we need to have stimulators placed into the motor pathways deep within the midbrain? Do we combine this with spinal stimulators? And there's now evidence to suggest that spinal stimulators could be beneficial in the stroke patient because it starts to restore some of the sensory information coming back from the spinal cord and can activate circuits. So anyway, we're going to see this move forward, I think. But even in spinal cord injury patients, where it's been a lot more heavily investigated in the clinic, the improvements are very expensive, so meaning that the kind of care that you need is very extensive. You need multiple, occupational therapists or physical therapists need to be present. We're trying to replace that with robotics, but it's a long haul, and the amount of care that goes into this with stimulators is something that requires months and months of effort. So the question is, how do we make this more accessible to multiple patients and tailor it in such a way that we can get an increase in improvement in recovery, right? And it comes down to these two questions. How does it work? And I'm going to talk a little bit about the mechanisms. And then why do we care? Well, the why is what I just explained to you. If we can improve the rate of recovery from electrical stimulation, we can expand this to more patients. If we can also understand better the mechanisms, we can add adjuvant care, pharmacological care, other methodologies to make the system work better and faster for patients. So that's the goal of understanding better. And technology better. So I introduced the first mechanism, which is the classic reason or mechanism we think for neuroelectrical stimulation, and that's Hebbian plasticity. There are a couple other mechanisms that are being explored. The second one really is priming of spared circuitry. So I mentioned most patients have some spared circuitry, and when you stimulate you stimulate those systems, you can bring them. You don't actually fire those systems. So what we do in the spinal cord, we don't actually fire the neurons. We raise their activity level, so we just bring them up. We tickle them so they want to fire. So this is priming a spared circuitry. And what this can enable in the stroke patient is it allows the patient to participate in rehabilitative care. So sometimes if a patient can't use the hand at all, if you can prime the spared circuitry a little bit, you can then allow some volitional control of the hand, thereby enabling care, enabling rehabilitative care. And that's an important concept for the application of electrical stimulation. And second one is immunomodulation. We know that in acute stroke, inflammation is a problem, but chronically it's also a problem. And in some of the early data, I'll show you just a little bit of this data, electrical stimulation appears to function partly through immunomodulation. So one of the pathways that I'm going to describe to you, I had two choices here. One, I could give you a whole bunch of data on sort of transcriptomic data on what happens when you stimulate the nervous system. And there is some of that out there. It's very little really in the general literature. Instead, I wanted to just give you an idea of the promise of trying to understand what happens when we electrically stimulate the injured nervous system. So this is just one gene. It's a gene that we've seen multiple times. If we stimulate cortically, if we stimulate in the spinal cord, one of the genes that gets activated within an hour of stimulation. So in an injured patient, in an injured rodent, you see this gene. It's called NR4A. And NR4A is a nuclear orphan receptor. It's a receptor, because it's an orphan receptor, it doesn't have a natural ligand. So people have often wondered, how does it get activated? And this data I'm not showing you, but data that has been repeated multiple times now, it appears that NR4A is a sensor of changes in neural activity. And why is NR4A interesting, and what could we do with it? Well, as you'll see from the arrows down below, NR4A doesn't just get activated in the neurons. Often when we do electrical stimulation, we think, well, we're activating neurons, and we're acting on this specifically. And we are. NR4A is in charge of multiple growth mechanisms in neurons, so growth factors such as mTOR and EGFR and c-MYC. And NR4A translates electrical activity into those growth cues that you see within the neurons. And that makes sense. But another thing that NR4A does is it actually shuts down, it decreases glial inflammation, particularly by astrocytes, and that's the second lane you see there. And one of the enigmatic things that NR4A does is it activates the neural immune cells, the microglial cells in the brain, and it upregulates their activity. And we think that it upregulates their activity in a way in which it helps these cells participate in the reorganization of synapses and or pruning of synapses that aren't working. So this is a single gene, and why it's important, it's a druggable target. And maybe that we can activate this gene in the context of neurorehabilitation and in the context of electrical modulation to try to increase the activity of these neurons that are activated or increase their regenerative programs. So this is just the tip of the iceberg. I think it's remarkable to me that in 2022 we still don't really know what the main transcriptional pathways are for electrical stimulation. And this is just the beginning. I think multiple investigators are going down this road. We're very interested in this particular pathway, but there's multiple that we can pursue. And I'll end just this part, and I'm going to end with again, a future technology that we're really excited about. But just to give you some other context of why stimulate. We know that electrical stimulation for the peripheral nervous system actually makes the peripheral nerve grow faster. So we need to understand how we can do that and apply that technology. And as I mentioned, we're now learning that electrical stimulation, particularly of the vagus nerve, can be used to modulate the immune system, which probably has important ramifications for the stroke patient. So the last thing I want to tell you about is in the stroke patient, we know that there's cell death. And so for fulminant repair of the stroke patient, we have to do more. Electrical stimulation is only going to take us a certain degree, even with these adjuvant therapies. So we know that there's a dramatic amount of cell death. That cell death can occur either in the descending input, right, in the cortex, subcortical structures. And what we need to do is we have to have methodologies to replace those structures. We know that. We can do that perhaps with electronics. We might be able to do that with synaptic relays, brain-computer interfaces that can act as relays across the lesion. But I think we want to look at ways in which we can provide a physical substrate for the patient that could replace the lost circuitry. So I'm going to show you, this is a project that we've published a series of papers on. And one of the challenges has been, as many of you know, people have been interested in using stem cell technology to create replacement cells for the nervous system. But the standard approach up until recently has been to make stem cells in a dish and or even make immature neurons and then inject those as a suspension and then basically hope that nature takes over and those neurons are integrated properly. So what I'm going to describe to you now, in our laboratory and multiple laboratories, we're trying some new technologies, and the idea is to look at the circuitry that's been lost. So what I'm showing you is this particular approach is being applied to the concept of motor circuitry, particularly motor columns of the spinal cord. But you can apply this to pretty much any other system in the brain. And what we're doing is we're trying to, in a dish, engineer that circuit that needs to be replaced. So in the top panel on the right, what you see is we take a skin biopsy of the patient, and then we engineer their cells to become neurons in a dish. So this is a cell engineering technique which is now commonly applied, and we can make different types of neurons in the nervous system, and we can make the corresponding glia. The unique thing that we're doing recently is we're using a technique where we encapsulate neural networks, and we allow development to occur within these encapsulated neural networks. And the compound that we're using right now is called alginate. So alginate is It's a polysaccharide that's found in algae, and it's a very versatile tool that we can make any kinds of shapes of neural networks. In this case, we're making these long tubular shapes, or what we call ribbons. That is that recapitulates what you see in the human spinal cord. And then we can take these ribbons, you can literally pick them up with a forceps, and you can show that they are functionally active. And then our concept is you'll be able to hand these to the surgeon, and the surgeon will implant those into the area where we need this new circuit. So what do they look like? Up close, you can see on the left, this is a neural ribbon. This is a motor column. So you see the red cells there are motor neurons, and they're we can make them very specific to the segment that we want to fix and repair. And the same thing we can do in the brain. So we can pick out the layer of neurons we want to make, and we can create them. And then on the right, what you see is that they mature, but more importantly is on the bottom. What you're looking at is field recordings from these neural networks. So this is ex vivo. And you can see the neurons are communicating, so all those ticks are neurons that are firing in clusters and communicating. And then if you spritz a little bit of an excitatory neurotransmitter on there, you see they go crazy off to the right from the glutamate addition. So these are functionally mature neurons, and we have multiple markers showing that these are differentiated neurons that are integrating into what would be a motor circuit ex vivo. Now if you take that and you inject it, so on the left you see this is an injured spinal cord, and that little black zone, this is an MRI post-operatively of injecting the ribbons. So these can be, we can put a contrast agent in the ribbon, and the surgeon then can implant them into the motor column, and that's what you see there, placing them surgically into the motor column. And on the upper right, this is the worst environment for these cells, right? So one of the challenges, this is a lesion cavity. So this is an area of the spinal cord that's basically had a tremendous amount of blood extravasated here. It's a cavity that's very inhospitable for transplants, but there's something about the alginate and these pre-differentiated circuits that appear to be quite happy there. And you can see that region where you see the red and the green, those are different types of cells within the ribbon. They survive quite well. And then the bottom panel shows you looking down the barrel of one of these ribbons in vivo. And the last panel is showing the number of synapses within that ribbon. So we're just at a point now showing that these ribbons survive, and we can put multiple ribbons, the surgeon can cut them to the lengths that they need, etc., and put them in vivo. So we're very excited about this technology, and I think the future really is combining this technology with neuromodulation, right? So once you get that circuit into place, then you need to be able to use Hebbian plasticity, right, to drive the new connections from the host into this grafted piece of their own nervous system. So I'm going to end there, and I think that the future from the bench is researchers like myself. We really want to develop a deeper understanding of how neuromodulation works, so we can provide more tools to the clinician to make neuromodulation accessible, have more rapid and functional effects for the patient. And finally, I think not so far off, I think the near future isn't that it isn't as futuristic as it may sound. I do think we can begin to think about designing circuitry for the patient and creating those circuits ex vivo from the patient's own cells and then implanting them. So thank you very much.
That's very exciting, Dr. Horner. It does really seem like we're at the doorstep of a paradigm shift in this area of neural recovery and restoration. And a lot of the work that you've done in your career centers around neuronal and glial regeneration. You've talked about implanting new circuits. We're involved here clinically at Houston Methodist in a major phase 3 randomized clinical trial pivotal study of stem cell infusion for stroke recovery, which is the MASTERS-2 trial. And then, of course, you've talked about many of the exciting neurostimulation modalities that, again, we seem to be really right at the doorstep of. I'm going to ask you to try to read the crystal ball, if you will, and among all of these potential advances and breakthroughs in neural recovery, which do you think is going to break through the first? Do you have any sense of that, and what do you think our next few steps will be?
Well, it's a great question. It's a tough one. So the crystal ball, I mean, I think one thing we didn't talk about, or I didn't talk about, but will probably be discussed today, is imaging. I think that one of the things that we need to know about the patient is exactly what their deficit is and also what their spared pathways are. And those spared pathways aren't obvious, right, because they can't come online. And so I think imaging combined with some exciting new technologies for stimulating the patient is going to come online so that we can begin to understand where these spared pathways are. And I think that will give us an opportunity to do very exciting designer electronics to stimulate in the right way, in coordinated ways. So it's going to be combinations of brain-computer interfaces, spinal interfaces. I really think that's a near future thing, a breakthrough technology that we'll see in patients. Right now we're kind of stimulating, and it's almost in the hands of the therapist really, stimulate and see what works for the patient. I think that once we learn how to map create functional maps for the patient, then we're going to be able to really go in and justify going in and implanting the appropriate technologies. And this is probably going to be expanded into robotics as well, right? So I think we're sort of dreaming this, the researchers are dreaming of this, but it's certainly within the realm of possibility that we could do this, where we could have muscle stimulators within the same patient, a spinal stimulator, cortical stimulators. The technology is getting really advanced. The devices are very small. In fact, there are now devices for electrical stimulation that are digestible so that they will disappear over time. They'll be absorbed by the body. So I think it kind of comes down to, do we just throw these things into patients and hope they work? I don't think we're going to do that. I think one of the breakthrough technologies and really is really doing some very sophisticated analysis of each patient and understanding where the opportunities lie to improve their function, right? So that may be, I think, maybe it's somewhat pedestrian, thinking of mapping the fiber pathways initially as the most exciting breakthrough, but I think it really is. It's really going to be very important and will shape how we care for the patient.
Thank you. Thank you for that stimulating talk.
Thanks.